Thanks very much, John. Can you hear me, or I wander around like this? I don't like standing at the lectern all the time. When Robert, I think it was, rang me a few months ago, seems a long time ago now, to say, you know, could we have a presentation? Would it be okay to think about how disease challenges perhaps older sites? You know, there's a lot of older free-range sites in the UK. You know, what effect does challenge have on these older sites? Could you possibly sort of think about that? And so we went through a few ideas about what we might talk about, and it seemed a good idea. And of course, it becomes so relevant in the last fortnight, doesn't it? So a lot of what I'm going to say is extremely pertinent, I hope, to the current situation. I'm going to mention avian influenza, the dreaded word, but I guess it's probably time at the end for questions would be a pertinent time to discuss a few things if producers do have concerns, and I'm sure we all have concerns. So what I thought I'd do is think a bit about, you know, what are the current challenges on older sites, and they can be split down into viral challenge and bacterial challenge, and as a poultry vet they're of incredible interest to me. And there's been a lot of work done over the years in terms of trying to recognise which are the important ones or which are not so important. But we've not really sort of thought about, and it's interesting, you know, Phil was just finishing off there talking about turnaround. Martin asked the tricky question, how can you do it in 3 days? You can't, can you? You can't. You can't get rid of the build-up of challenge in that sort of length of time. And I suppose if Phil and I were going to sit down and argue the toss, there'd be one commercial viewpoint on one side and a biosecurity and disease challenge viewpoint on the other. So it's a very difficult thing to get a handle on. And we don't really measure build-up of infection in free-range units. We do more in broiler units than we do in free range, but we don't really measure what sort of build-up there's been. So what I thought I would do is just focus in, and we've got 20 minutes and it's not a lot of time to talk about veterinary disease matters. You could spend all day, and I'm sure you'd all want to spend all evening talking about veterinary disease matters. But we've got half an hour, so I thought I'd focus in on just a few areas, namely some of these unexplained egg drops that we come across, which are extremely frustrating to try and get to the bottom of. Increasing mortality problems can be very complex in how they manifest themselves and what the causes are. And following on from what Phil was kind of just started the ball rolling, I'd like to just talk a little bit about some work we did with ranges and what potentially can happen there. So let's just set the scene. Intensive versus free range. Massive difference in terms of the birds and how they're housed and the environment that they're in. We all know, you know, the difference is free range birds have got contact with the range, or some of them have. How many birds go out in your flock? People reckon it's probably only about 25% of an average flock actually go outside, but there is range contact. Therefore, they have contact potentially with wild birds. There's that trigger, H5N8 starts springing to mind. Direct effects of weather must have a huge impact on the way birds perform, and obviously water sources will also be a concern. Single age versus multi-age. I don't know how many of you have multi-age units in the room. There are still quite a few dotted around the country. But they're not easy to manage from a disease point of view and a biosecurity point of view. Some of the biggest issues I've come across as a poultry veterinarian in this country are usually multi-age sites, and trying to figure out how you're going to address stopping the transmission of disease from one unit to another, or control vermin, which will inherently move from one site to another on a unit, is very difficult. And lastly, how do we control vaccination? If we are going to talk about vaccinating on the farm, I'll mention that again in a minute, how are we going to do it when you've got a multi-age unit? It's quite complicated, it's not easy to do. So let's just focus in a bit on some egg production problems, which I have to say are probably one of the commonest things that we get called in as a poultry vet. I'm sure the other practices would have something similar. You know, Egg drops, commonest problem we come across really, which vary tremendously from a very slight blip in terms of 1 or 2%, not quite sure why they're dipping, but they are, down to quite a major crash of 20 or 30%, and can be extremely damaging to the producer and quite frustrating to try and get on top of, because the diagnosis can be difficult. What I would suggest as a general approach here, and I'm just going to give you some ideas maybe as to what you might be doing in terms of managing the birds and possibly some proactive things you could think about, is looking at, we've relied on things like antibody levels in the birds and taking blood samples to look at antibody levels. Maybe there's something else we can do there to try and give us more information. And maybe in this modern age, we can start thinking about some quite up-to-date techniques to try and find the virus that's causing the problem. Could we take swabs from the birds, pop a swab down the throat from the rear end, so tracheal and cloacal swabs, and actually use good techniques to find the virus that's causing the problem? It used to be a purely experimental technique. It was expensive, it was time-consuming, and it wasn't something that we could do in the real world, whereas nowadays we can. And you'll find that the vaccine supply companies offer an excellent backup service through the vet to try and identify the potential problems that are occurring on a unit. So if we want to try and find out if IB, ART, or possibly even low pathogenic avian influenza is causing a problem with egg production, then that's something that we should be thinking about. Just on that point, 2 weeks ago, there was the first outbreak of high pathogenic avian influenza in East Yorkshire. One of my colleagues was called out, I think it was a Thursday now, can't remember, it's like Thursday or Friday, was called out to the site because there'd been an egg drop. And it was an egg drop that at that stage was quite severe. But we'd had other egg drops over the years that were probably something similar, and were probably due to management problems or there were specific reasons. Anyway, he went out and looked at these ducks, these breeding ducks that I'm sure you're all aware of, and found that the ducks looked relatively healthy. Yes, food consumption had gone down, and there was a huge crash in egg production. 5,000 eggs down to about 500 or so on that day, so a massive crash. And he was thinking EDS, which we know can be a problem in ducks, that's a possibility, another viral challenge. I'm going to have to probably report this to the ministry because it could be avian influenza. The rest is history. But it was thankful that that was a breeding flock that he went to see and not a commercial grower flock, because if it had been a commercial grower flock, I'm pretty sure he wouldn't necessarily have had avian influenza at the top of his list, because the birds looked inherently not too bad, and it was the egg production that had crashed. So, food for thought. Species. What breed have we got here? Can you see at the back? Any ideas? Turkeys. Correct, turkeys. And I'll give you a clue. There aren't many left alive in this shot. There's one here, there's one there, one there, one there, maybe one there, but they're dotted amongst the shed. That was in Holland, 2003, avian influenza. 12 hours after he shut the shed up, he opened it up and that's what he saw. So that was that level of mortality in 12 hours. And that was a different virus. That was H7N7 back in 2003. Very high mortality. And unfortunately, turkeys die in large numbers with avian influenza, closely followed by chickens. Ducks, hardly at all. Massive difference between the species. So if we're thinking about avian influenza, and we see that, that's very typical. But egg drops, you don't tend to think about it as much, but that's what we saw a few weeks ago, and potentially that's what you could see on a farm, not necessarily birds dying in those numbers, but you'll certainly see drops in egg production. So it's just something to bear in mind. The normal reason for egg production drops is IB. That's the commonest organism that we tend to see. And if we'd been speaking about this organism 20 years ago, we would have probably been concentrating a lot on the clinical signs that we see in the birds in a flock. We'd have seen discharge from the eye, we'd have probably seen discharge from the nose, we'd have heard a cough, a snick, a head shake. Very, very common. And yet, nowadays, yeah, we do see those occasionally, but not as common as they used to be. What we tend to see are egg drops and poor shell quality. So there's been a real change in emphasis in terms of how these viruses present themselves over the last few years. If you were all broiler producers, we'd be all talking about weight gain rather than respiratory signs. Similar scenario. So these viruses go right around the body. They're not just a respiratory virus, they affect the gut, the way food is absorbed, they affect the reproductive tract and how eggs are produced. And these are some of the signs that we tend to see from challenges. There's a research worker in Liverpool, Dick Jones, who's probably the world expert on infectious bronchitis. And he was able to show that these viruses, and we're talking the last sort of 20 years or so, all these different strains of IB that have come along, you probably know the names of some of them, 793B, D274, 388QX, horrendous names. I don't know why they have to call them that, but that's what they do. And he's found that they probably almost certainly originated from the top of Africa and then migrated up either on the wind or via people or via wild birds potentially into the Mediterranean countries and then into Northern Europe and certainly into the UK. And that seems to be the sort of the route that they've traveled. So over the sort of last few years, we've come across lots of these different viruses causing challenges. And we've taken blood samples and looked for antibody levels to these viruses. And it can be really complicated and very frustrating to try and interpret results from a blood sampling exercise. And these were some results that were, I mean, this is from a few years ago now. I just grabbed one out of the record, changed the name and the address, and put them on the screen here. But you know, you've got antibody levels there to 793B that are moderately positive. You've got not quite so high to 1466. You've got D274 down here. We've got 755 there, and we've got 388 at the bottom, all showing positive levels in kind of in the middle range. How do you interpret that? Really, really difficult for me as a poultry vet. If you presented, or if we found these results on your farm, how do we interpret that at all? It's really difficult. So I would suggest that we kind of just need to add a little bit of information into the system so that it makes it easier. And I think the way forward is to take regular blood samples through the laying cycle that you guys experience. And I would recommend that it's worth taking samples at regular basis that you don't necessarily need to use, but you can go back to at a later date. I'm sorry, those arrows have just gone out of sync slightly when we reformatted the presentation. But the idea is that we possibly take some blood samples when birds first arrive. This graph is just very graphically to show an average antibody level that might occur following vaccination in rear and possibly following the inactivated vaccine or possibly a challenge on the farm. It shoots up to high levels. So if you can take blood samples on a regular basis, say at 20, 30, 40, 50 weeks, you don't necessarily need to do anything with them. You certainly don't need to sample all those blood samples and test them, but you should certainly take them and store them, and then you can go back to them at a later date. So if you get a challenge at 50 weeks, you can go back to the 40-week sample, pull them out, take some more bloods a fortnight after the challenge, and you've got two samples relatively close to each other that you can compare antibody levels. And that way, going back to that graph that I put up before, that table of results, we can compare antibody levels to see if they're going up, staying the same, or going down, which will give us a clue as to which virus is causing the problem. So what I would recommend that producers do is take regular samples, send them into the veterinary laboratory, spin them down, we freeze them, don't charge for that. we might throw them away at the end of a couple of crops, but you've got the data to go back to if you need it, and you can then retest at a later date. In conjunction with blood sampling, I would always recommend that people try and work with their vets to find out which viruses are causing problems by taking swabs. So using tracheal swabs and cloacal swabs together to look for the DNA of the virus with a PCR technique in some specialized laboratories. But as I say, the vaccine companies are really good at supporting that sort of analysis. And it's information that's really, really useful if we're going to make progress in terms of what we're going to do. Phil mentioned that he actually vaccinates on his farm because we recognized that there was a problem on Phil's farm with a specific IB virus. So although we are vaccinating with a very comprehensive program in RIA, and it does seem sometimes amazing that we can get so many vaccines into a pullet that we do at the moment, and we're trying to sometimes even give more, which is difficult. But we give a combination of IB vaccines through the rearing process. We finish it off with an injectable vaccine at transfer. And then it is possible to vaccinate on the farm. But I'm going to put a caveat there. We would never, ever recommend that people vaccinate on farm unless you've got extremely good evidence of a specific field challenge. So if we find there's evidence of a 793B virus or a 388 virus, then fine. We'll think about a program, and it may well be the appropriate thing to do on the farm using the right bit of kit. But we don't go down that route unless we've got good evidence that it's going to help. If you are going to vaccinate, use the right bit of kit. We tend to recommend, certainly on the flatbed systems, Ulvavac sprayers are excellent. They're excellent because the technology has been developed to produce an extremely consistent droplet size. So the spinning disc that goes round on the top there has these little teeth, and when the vaccine drips out of the bottle through the nozzle onto the spinning disc, it produces a very, very consistent droplet size. Not small enough to get right into the lungs, but small enough to get into the upper respiratory tract, down the airway, and then it stops. But that's where immunity is produced in the bird. So you get a very consistent droplet size produced by that bit of kit. Now, I do understand the problems with multi-tier systems, and vaccinating those particular houses offers a challenge. There are other types of kit to the Ulvavac which we can use, but I would always recommend that people vaccinate against IB using a sprayer rather than through the drinking water, because you'll get far better local immunity produced in the bird. So in my opinion, I think most opinion of the vets in our practice, we would always recommend trying to spray. Okay, let's change subject slightly. We're on track. Mortality issues. Very frustrating, again, to get to the bottom of some of these issues. And we're seeing a lot of scenarios where we're seeing gradual increases in mortality throughout lay, often associated with quite severe stress factors in the flocks. Lots of different causative agents, Erysipelas and E.coli probably the most important, though we're obviously seeing mycoplasma issues as well from time to time. Extremely limited options in terms of what we can do about it, because we've only got a limited number of products that we can use with a nil egg withdrawal or a licensed, and extremely challenging to get to the bottom of it. So I would suggest that rather than trying to bang antibiotics, and yes, this is a vet saying we should use less antibiotics, we should look at preventative strategies rather than fire brigade strategies. I'll just go through a couple for a minute or two. Erysipelas. Phil was a pig farmer before he went into chickens, and we had a conversation on a chairlift in a poultry on the piste a few years ago, and he said, should I be vaccinating against Erysipelas? What's the issue here? And the answer is most definitely, because Erysipelas hangs around as spores in the soil, and will hang around for long periods. So people who have had pigs even 10, 15 years before and have been out of pigs for that length of time We still see birds going down with Erysipelas. It's a big problem. So yeah, it's a sort of pattern that develops over the period of the flock, and you'll get a slow progressive increase in mortality. But we're starting to use more and more autogenous vaccines against Erysipelas. Store that one up, I'm going to come back to it. E. coli, the commonest environmental organism that we have, lives in the gut of mammals, of humans and birds, lives in the environment and is a common environmental organism. You know, when we're talking about cleaning sheds out, E. coli will be an important part of the makeup of the bacteria that are in those houses. And huge reservoirs in wildlife, in soil, in water. So it's a very common organism. What sorts of clinical signs do we see in birds? Well, they vary tremendously. We can get respiratory signs because of E. coli. So you can get coughs and snicks and rales and swollen heads and sinusitis purely from E. coli infections. We can get problems associated with E. coli causing drops in egg production. So often when we're seeing episodes of peritonitis towards the end of the laying cycle, we will often find E. coli associated with them. Now, are they the primary cause or a secondary cause? Sometimes difficult to tell, but they're certainly important. And they're probably one of the main causes of mortality in free-range flocks. So I thought it was only right, as a poultry vet, we ought to have some good post-mortem pictures. You've had your lunch, so okay, here's a, not too bad, dead bird on the PM room table. But yeah, common findings. We will often see peritonitis, where you get this buildup of fibrin-like material where an egg has broken, becomes very inspissated through the body cavity, and then certain changes start to take place. Something that we see an awful lot when we're post-morteming laying birds. But what are we going to actually do about it? Well, yeah, it's important to try and find the primary pathogen, the organism that's causing the problems. Is it E. coli? If it is E. coli, is it a nasty sort or a not so nasty sort? There are different forms of E. coli. There are many types of E. coli, hundreds of types of E. coli. So finding out which one it is quite important. Is mycoplasma involved? Is IB involved as a primary trigger, and then we're getting these bacteria in on the top? Or maybe it's a feed-related problem with mycotoxins. Or is it just the management in the shed or contamination of the water? There are so many factors that could be involved when we're talking about E. coli infections. A lot of people just think, well, it's okay, we'll just need antibiotics. And yes, we do use antibiotics to treat those sorts of infections. But I hope that we normally at least try and culture them and maybe look at sensitivity testing to see which is the most appropriate antibiotic to use. Bearing in mind we have 3 or 4 licensed products with nil egg withdrawal, but only a proportion of those are probably going to be effective against E. coli. So we're really limited in what we can use. And I know a lot of people in the room will probably have taken the approach, well, we should put an antibiotic in, colistin or an oxytetracycline product that's licensed, and let's at least treat them for a week and see how we get on. We used to rely on Orafac, didn't we? CTC in the feed. That was our standard sort of treatment for these E. coli infections. Well, I would suggest that in the short term, maybe you can use them, but in the longer term, we've got to think about alternatives. And we've got to think about maybe eliminating the other causes, which would include vaccination strategies, thinking about managing the birds better, making sure that we do worm on a regular basis and make sure that worms aren't playing a part of it, making sure the management of the house is correct. And probably more importantly, I think Kelly hit on, she mentioned water systems this morning, and Phil certainly just mentioned, you know, keeping his water system clean. But unless our water system and water administration is done correctly, we don't stand a chance. People always ask me, yeah, but it's okay, you know, clean the water, but they're drinking out of all these puddles outside. Well, that's fine, and yes, they do, especially when there's been a lot of rain, but it's only a proportion of the birds that go outside to drink. And secondly, if the bulk of the water that they're drinking is from the in-house water system and it's not clean, you're on a hiding to nothing. So administering the water system correctly is really, really important. I mentioned before there's loads of different types of E. coli, and they're all classified with different sort of categories based on the structure, the cell wall, the membranes that they have, and these flagellae that stick out from the surface. So there are lots of different types. Some are good and some are bad. Actually trying to type those and figure out if it's a good or a bad type is really, really important. And we've increasingly become reliant on autogenous vaccines. And I mentioned it also for Erysipelas. The same thing applies for E. coli. And we're using a lot of autogenous vaccines to try and help combat some of the problems that we see. For those of you in the room that aren't familiar with autogenous vaccines, they're basically a bacterial, it could be a viral vaccine, could be a vaccine against a spirochete in the gut. They're possible as well, but it's usually bacterial, and they're made from the specific pathogen that's been isolated from the flock of birds on your farm. So it's a vaccine that's specific to your site. They can only be made under license, and they can only be administered and delivered under license. So they're a specific POM organism, vaccine. So they've got to be made on premises that are licensed. So you can't just, I couldn't just go into into the back of the PM room and start rattling up an autogenous vaccine. It's got to be done under strict conditions, which are licensed by the Veterinary Medicines Directorate. We use Ridgeway. I know Tim's here at the moment. He's got a stand, and they're very effective at supplying the autogenous vaccines for use in the UK. And I think we're indebted them to a large extent for that. And they're targeted against specific pathogens that are going to be a problem. So by the very nature of them, they've got to be delivered in a certain way to each bird. And because they're inactivated, in other words, the organism's been killed first and put into a bit of a pot to make this vaccine, they've got to be delivered by injection. You can't spray vaccinate an autogenous bacterial vaccine. But we've found them to be extremely effective in reducing mortality, assuming that bacterium is the main cause of the mortality that we're seeing. And that's an important point. So it's part of the vaccination program for the next flock in Rhea. I once did use an autogenous vaccine in a flock in Ley many, many years ago. It's not something I would ever do lightly because of the stress incurred on that flock when you actually vaccinate them, picking them up, jabbing them. It's not something we would do normally. But it's normally for the benefit of future flocks. So you're thinking about putting it into the vaccination program in future. And you've got to plan this because it takes ages to make a vaccine, in the sense that you've got to find the organism, purify it, you've got to grow it, you've got to inactivate it, you've got to make the vaccine, then you've got to test it, and you've got to do an on-farm safety test before you deliver the vaccine to the birds. So if you've got a 65-week flock and we've just grown a new bug, we're going to really struggle to make a vaccine in time for your next placement of pullets. Before 50 weeks, yeah, we stand a chance. If it's just a repeat vaccine, it's a much quicker process because Ridgeway will almost certainly be storing samples of the vaccine that they can just grow up and produce. So it's time consuming is what I'm saying, and it's not the sort of thing that you can undertake just on a whim. But in addition to the use of autogenous vaccines, you must increasingly be thinking about the quality of the water and using products to reduce the chances of bacterial contamination going into the birds, using products like peroxide, organic acids, chlorine dioxide, the sorts of products that will remove biofilm and therefore get rid of the bugs that are potentially in your system. And nipple systems are no different to bell drinkers. They will get contamination inside the system very, very easily. So we would always follow up a treatment with an antibiotic on a problem with E. coli by sanitizing the water where possible. And hopefully the combination of those two will produce some good results. To finish off with, I just wanted to talk about the range. Phil started to talk about the range and what he does obviously on his site. But we don't really know how much importance the range has. We suspect that it's probably a cause of contamination for the birds, but we don't really know. There's very little data at the moment available to find out whether it is a problem or not. It's certainly weather dependent, and tremendous variation in what people do with the range, whether they put stuff outside the pop holes or not, and what can we do really about providing solutions. So we did a little bit of work, and in order to do this work, we had to sort of get some friendly clients and get to know them really, and figure out what we were doing. So we got to know our clients, and there's Phil practicing actually I don't know if you're aware, but he's in Strictly in 2015. So this is him practicing for that. You've seen this slide before. This is Phil's site, which, okay, with the birds present, with the birds, you saw this slide before, but you can see here that it's quite challenging in terms of we've got a concrete area here, we've got a stone-filled area here, we've got paddock that's starting to regenerate, down the length of where the birds can go. Phil has a paddock rotation system, but not everyone does. But this is probably pretty typical of the sorts of unit that we see. And obviously when the birds have been in it for some length of time, as Phil quite rightly said, it gets poached, it's a mess, etc. So what we decided to do was to try and measure soil samples, take soil samples and try and measure the bacterial contamination every 5 meters on a strict line down the range. And it was quite interesting to see the sorts of results that we got. We did it over the course of about a year. We chose 2 free-range sites in Yorkshire, allied to a particular packer. And we took soil samples from a roughly the size of an A4 sheet of paper, about a couple of centimetres deep, and we analysed for worm counts, cocci levels, not that we expected there to be many cocci, but certainly we were interested in bacterial levels. We also took samples from the garden at the practice. I sampled a couple of sites because I didn't know what the average bacterial level of an A4 sheet of size bit of soil, what the bacterial level was. Hadn't got a clue, never measured it, didn't know. So we did that as a bit of a control. And it was quite interesting to see that, first of all, the worm counts were incredibly low all year round. Now, Phil and the other producer do worm religiously, so that was good. Worm levels were low. When we looked at control samples, they were between about 50,000, 200,000 colony-forming units per gram of soil. That means bacteria, to all intents and purposes, 200,000 bugs per gram of soil, and a gram is about the size of the end of your thumb, so to give you a rough idea. But it was very interesting when we followed those samples down the range that we found a tremendous variation from about double what we were seeing in the control samples up to 25 million per gram of soil. So huge increase in numbers. Now, we didn't go down the route of trying to identify all the bacteria because it was a big soup of bacteria. But it just gave us some ideas as to what sort of contamination there was. And in general, bacterial levels, I suppose, as you would expect, dropped the further away you went from the pop holes. So that was sensible and probably what we would expect. There were 1 or 2 little blips. If the water had accumulated around a sampling point, it was generally higher. But in general, it went down. And the significant area was the first 15 meters of the pop holes. That's where the levels were highest. And they dropped very quickly after about 15 meters. So it seemed from this very simple, and it's not, I'm not putting it in a scientific journal or anything, but from that work that we did, it seems like the first 15 meters is critical. Interestingly, the area of concrete that Phil had, he's got about sort of 5-6 meters, I think, width, roughly, of concrete had incredibly low levels. So the washing effect of rain and probably the fact that he could clean that more effectively anyway meant he did get rid of the bugs there. So that was good. And also, winter samples were much higher in their level than summer samples. And I suppose that's what you'd expect as well, because of accumulation of water, maybe you get washing of bacteria into that sort of area. So yeah, That was the sort of level of result. Just to finish off, Mr. Chairman, what can we do about it? Well, I think a few thoughts. We should certainly keep concrete areas, if you've got them, very clean, if you can, just outside the potholes. Make sure that the whole area is well drained, something that sometimes we don't always think about. We've tried using things like bark chippings outside the potholes, and if you haven't got concrete, something like bark chippings appears to be relatively good at providing good drainage. You can replace it easily, it keeps the birds off the ground below, and therefore we think it probably reduces challenges. And keep that replaced and refreshed on a regular basis. Remove droppings from the range, common sense, harrow the grass. Get the UV light at the grass, and that will have an effect on bacterial levels as well. And I started talking about avian influenza, and obviously that's very pertinent at the moment. Let's not forget that it is all down to biosecurity. And biosecurity is not rocket science. Biosecurity is common sense that we all should be able to do. So stopping people coming on, using proper equipment to protect people and protect the birds from people. So in terms of clothing and boots and everything else, vermin control, and yeah, okay, wild bird control is tricky on a free range production, but have you got standing water? Can you try and keep wild birds away if possible and keep animals away? There were some of these outside the door when we walked in today, really good type of foot dip. Brilliant, because the good thing about those is, I would encourage you all to get one, and I haven't got shares in them honestly, you can measure the concentration of products that you put in there. It's not just glug, glug, glug in the bucket, top it up with water, that will probably do. It's actually the correct concentration to kill the bugs in the first place. And don't forget hand hygiene. Just as important as the boots are these things. And we can pass infection on so easily. I think more important probably than overalls are hand washing and boots, to be perfectly honest. Though maybe you should be doing all of them. So, thank you very much, Mr. Chairman.